So I have no financial disclosures for this talk either. Just a little background. Current gene therapy trials deliver gene therapy vectors by either subretinal or intravitreal route. The subretinal route is the most common one. We can go to the next slide. And the commonly used vectors are adeno-associated virus, you could go to next, or lentivirus. Now, these vectors do not penetrate the retina well, and they have to be delivered subretinally if photoreceptors or RPE cells are targeted. Here I've listed, next slide, the major current retinal gene therapy clinical trials. Most of them are phase 1 or 2, and you could see Stargardt's, Usher's, X-linked retinoschisis, choroideremia, achromatopsia, exudative AMD, and retinitis pigmentosa trials going on. Now we have the first phase 3 trial for gene therapy. It's RPE65, Leber congenital amaurosis or retinitis pigmentosa by Spark Therapeutics. Hold on for 1 second. The gene therapy for RPE65 diseases is expected to be released by FDA soon. Bascom Palmer treated patients within choroideremia and achromatopsia trials, and this is the experience that I'm going to show with you here. Now, this slide that you're seeing right now shows subretinal injection instrumentation. Please hold on, do not, can you go back 1 slide? So these instruments, subretinal cannulas, are 23 gauge instrumentation, and they taper to 41 gauge extendable or fixed cannulas that the surgeon has to insert subretinally. So that's the key, make sure that you're under the retina. Next slide. Here's an example of blebs, they're outlined in yellow, from Professor McLaren's choroideremia trial, where he used balanced salt solution to raise the retina, which is very adherent to the sclera in choroideremia patients, and it was not easy. It's a really difficult step, so making sure that we are under retina instead of under the RPE or suprachoroidal is key. So let's go on to the next slide. So I would like to show you what we did in our choroideremia trial that we conducted at Bascom Palmer Eye Institute. It was a phase 2 trial. We used intraoperative OCT to really make sure that we were under the retina. Here you can see subretinal fluid and suprachoroidal fluid pockets after balanced salt solution was injected under the microscope, and it was a Lumera microscope with rescan. So just looking at the retina, we really couldn't tell where we were, what plane. And here you can see subretinal and suprachoroidal fluid. We tried to increase the subretinal fluid pocket here, and we were unable. So next slide, we went to a different area, and there we were able to create a large subretinal fluid pocket into which then a cannula containing gene therapy was inserted, making sure that we were in the subretinal fluid instead of suprachoroidal fluid, so we could deliver gene therapy under the retina. Next slide. Here's an example of achromatopsia CNGB3 therapy phase 1-2 trial that we treated the patient within Bascom Palmer Eye Institute. And you can see, again, the subretinal fluid. This is balanced salt solution before gene therapy is delivered. It's nice, too, for the surgeon to know the plane that the cannula is within. Next slide. It's also handy to use the intraoperative OCT for monitoring for possible complications. Here, you could see an impending macular hole, which is not an uncommon occurrence within these trials when the retina is being stretched, especially with large subretinal volumes of gene therapy. So here we decided to abort further injection of gene therapy product, so we wouldn't create a large hole and express all of our agent into the vitreous cavity. So it's a handy tool to have. And in conclusion, next slide, there are multiple ongoing retinal gene therapy trials utilizing subretinal delivery of viral vector. Visualization of retinal layers in retinal degeneration patients may be difficult, so it's nice to confirm subretinal injection, and intraoperative OCT provides real-time feedback to guide viral vector injection and allow allows detection of complications such as incorrect layer injection or impending macular hole that would not be otherwise visible. Thank you.